Hello everyone, I'm Alisa Kumbara from the University of Verona, and I'm excited to present our work, CRISPR-HAWK, a toolkit for designing haplotype and variant-aware CRISPR-Cas guide. CRISPR-Cas has revolutionized genome editing. This system has three key components: the Cas nuclease, which performs the DNA cut, the single guide RNA, which directs the CRISPR-Cas complex to its target across the genome, and the PAM motif, which ensures target specificity. Designing an optimal guide means maximizing the on-target activity while minimizing the off-target effects. However, most current single-guide RNA design tools depend solely on the reference genome, ignoring that single individuals may carry genetic variants. These variants can impact CRISPR genome editing outcomes by modulating guide efficiency, disrupting the intended target sites, or even generating novel off-targets that would not be considered from the reference sequence alone. This is especially important in clinical and personalized applications. So a guide performing well on the reference genome may behave differently in someone carrying a variant in the target region. It might bind less efficiently, lose its cutting ability, or gain unpredictable off-target effects. Our tool CRISPR-HAWK addresses these challenges by making guide design aware of individual genetic diversity, so considering both variants and haplotypes to improve specificity, safety, and overall editing performance. First, we set out to develop a computational tool that can design single-guide RNAs while accounting for individual and population-specific genetic variants. By doing this, we aim to enhance the precision and safety of CRISPR genome editing applications, especially in personalized or therapeutic contexts. Second, we wanted to evaluate how well existing reference-based guides perform across diverse populations, potentially identifying efficiency limitations. CRISPR-HAWK works through four main modules. So first, the tool extracts the regions of interest from the reference genome using user-provided BET files. Then, CRISPR-HAWK retrieves genetic variants located within the input regions. The tool handles both single nucleotide variants and indels, supporting both phased and non-phased data, reconstructing all haplotypes for the following analysis. Then, each haplotype is binary encoded using a 4-bit scheme based on UPAC codes. In particular, this enables a fast and efficient search for CRISPR guides. CRISPR-HAWK scans these binary encoded haplotypes to locate valid PAMs, and then it extracts the adjacent spacer sequence. It flags any variants within guide candidates and provides detailed annotation for each spacer. We also integrate predictive models to score guides for both on-target and off-target activity. Then, finally, everything is compiled into a detailed tabular report. In particular, this includes guide sequences, their genomic positions, genetic variants, and functional annotations, together with some guides efficiency and specificity scores. To explore how genetic diversity impacts CRISPR guides' performance, we combine two scoring systems: the azimuth score, which predicts the on-target activity and the editing efficiency of each guide, and also the CFD score, which estimates changes in binding affinity and specificity when mismatches between the guide and the target occur. So together, they provide a comprehensive view of how reference-based guides behave across genetically diverse individuals. We applied our tool to the BCL11A erythroid enhancer, which is a clinically validated therapeutic target for sickle cell disease and beta thalassemia. And we used genetic variants data coming from both 1,000 genomes and the Human Genome Diversity Project dataset. We found that 30% and 40% of these guides overlapped with genetic variants. In particular, 4 and 2% of these were exclusive to non-reference haplotypes, meaning that they were created by novel variants-induced PAMs that would be missed entirely by reference-based approaches. We then focused on worst-case scenarios where guides that perform well on the reference show reduced predictive efficiency in variant-carrying individuals. So we used azimuth scores, and we ranked guides by the drop in predictive efficiency between reference and variant haplotypes. Several examples stood out. So in the 1,000 genomes dataset, the top 10 ranked guides showed significant azimuth score drops compared to the remaining pairs. In contrast, in the AGDP dataset, while efficiency reductions were generally less pronounced, the top 7 ranked guides still displayed notable decrease in predictive activity. For each of them, we tracked down how many individuals carried the efficiency-reducing variant. We also evaluated guide performance using CFD scores to quantify on-target efficiency across 1,000 genomes and HGDP individuals. Some guides, like those targeting the HBBB and FANCF in the 1,000 genomes, showed remarkable robustness, notable efficiency losses, with score dropping below 0.4 and 0.5 in many individuals. Instead, in the HGDP dataset, guides targeting the HBB and TRBC1 and 2 showed remarkable robustness, maintaining a high efficiency despite genetic variation. Notably, these efficiency drops were not accompanied by increased off-target effects, suggesting that while specificity remained stable, therapeutic utility could still be affected in variant-carrying individuals. In conclusion, our findings underscore key challenges for therapeutic genome editing in diverse populations. We found that 30 to 40% of the candidate guides overlap with genetic variants, and many of these guides showed reduced predictive efficiency in individuals carrying genetic variants. This suggests major limitations of relying solely on the reference genome for guide design. By incorporating population-specific variants, we can design guides that are more robust, reliable, and personalized, ultimately supporting safer and more effective therapeutic strategies. So our work suggests that considering population-scale genetic variation could be valuable for developing comprehensive therapeutic genome editing approaches. So thanks for watching. I look forward to discussing CRISPR-HAWK and answering your questions during the poster session.